We welcome everyone this morning remotely and right here in the room. It's with great pleasure that I welcome Karl Klontz. Before I get into his introduction, let's just pause for a word of prayer. Gracious God, we come before you with hearts full of gratitude for Karl, whose scientific insight and dedication to learning and knowledge and service have inspired us all. May we honor the gift of knowledge as a pathway for one another to serve the greater good. In unity and thankfulness, we offer this prayer. Amen. Amen. It is really with great pleasure that I introduce Karl, such a valuable servant leader member of our congregation. He's 40 years experience working for the Centers for Disease Control and Prevention, the U.S. Food and Drug Administration, as both a physician and epidemiologist. Karl's going to be updating us with the status of bird flu. During a career focused on infectious diseases, Karl has cultured the sewers of Miami for cholera, bled birds for encephalitis-causing viruses, served a sabbatical in Bangladesh at the International Center for Diarrheal Disease Research, and investigated multi-state and infectious disease outbreaks. For 17 years, he's also lectured part-time as a professor of epidemiology at the George Washington University of Public Health. Karl has traveled worldwide looking at disease outbreaks. He's also traveled worldwide serving third world countries with solar power, solar energy. He's worked with Clint Kelly in the Democratic Republic of Congo. Karl is one of the most remarkable people that we have as a treasure in our congregation, and we really appreciate his being with us today. So I will at this point let him take over, and then we will be seeing some slides from him, but go ahead.
Thank you.
Yeah.
With your permission, I'm going to stand near my slides. That way I can communicate with them as well as I can communicate with you.
Okay.
Join the two. So, Carol, we're okay with microphones and everything then? We're set to go.
Yeah.
All right, let's get that first slide up then.
Okay.
All right. Well, first of all, thanks for being here with me today to talk about avian influenza, bird flu. Its current status is what we'll focus on. Let's go to the next slide.
Okay, hold on. We've got some chats. Here we go. All right.
Okay, so I want to start with some good news. But before I get into that, I have about 25 minutes, 30 minutes of presentation material. That'll leave plenty of time for discussion at the end. But if there are questions, let me know. Let's do them as we go along so that we can not leave any loose ends here. So Carol, we have some sort of lock in Zoom. We can just X that out.
I'm trying to
yes.
actually get you in the screen.
Oh, okay.
Okay.
Well, let's actually, we'll just slowly start here as we work on that. So the good news is regarding the pandemic and the pandemic potential and what sort of preparedness, where do we stand in sort of preparedness should we have an H5N1 pandemic? And this good news comes from a federally funded group called the Administration for Strategic Preparedness and Response, or ASPR. And they lead the nation's medical and public health preparedness for, or response to, and recovery from disasters and public health emergencies. And as of now, at least, with regard to H5N1, ASPR concludes there's currently a low risk to public health. That's our present official status right now. They back up this contention by, I think, really important things we need to know. First of all, there are stockpiles of personal protective equipment, antiviral drugs, should we need them. and raw materials for vaccine production against avian influenza virus. These stockpiles include components of vaccines which induce immune system to protect against this virus, and adjuvants, compounds which reduce the amounts of ingredients needed for the vaccines, makes them more efficient essentially. Okay, let's go to the next slide.
Did that happen?
Should be the up arrow if it's on orbit. Let's see.
I'm sorry.
All right, we'll get that.
I don't know why I just hit missing. Maybe here. Here we go. There we go.
Perfect.
Sorry.
Okay, having said that, the preparedness for a possible pandemic. I think it's fair to say that the risk of an H5N1 pandemic is not zero. There's a possibility out there for sure. And I think CDC summarizes this very well in sort of precautionarily, quote, this is straight out of Morbidity and Mortality Weekly Report. The risk to the public might change based on whether H5N1 viruses acquire genetic changes that increase their transmissibility to and among humans, which could increase the risk of an influenza pandemic. That's a very good, honest appraisal of where we are. It's low risk right now, but it could change. So as such, today, what I want to do is focus on the current status of H5N1. I want to talk about biology and epidemiology so that we lay out sort of the facts of where we are. I'm not going to speculate on whether we will have a pandemic because I have no clue. I don't think anybody really knows for sure. So let's focus on biology here, get the basics on the table, and that way we can read newspapers and converse with science as our guide here. So let's go to the next slide and begin. So background. Two major types of influenza viruses circulate among humans, primarily A and B. These viruses cause seasonal epidemics in humans. Only influenza A viruses can cause pandemics, however. So influenza A viruses are established in many different animal species. For example, they're in birds, and thus they're named accordingly avian influenza viruses. Influenza viruses are in swine, that's a whole nother group. Horses, bats, cows, dogs, etc. And these animal influenza viruses are distinct from human influenza viruses and they don't easily transmit to and among humans, but they do cross species here and there. In a typical year, influenza A accounts for about 3/4 of the human cases we see, influenza B, the rest 25%. By the way, there is an influenza C and D, but very rare. Transmission, as we know, of influenza is through the respiratory route. So coughing, sneezing, touching contaminated surfaces will do it as well, transmit infection. Common symptoms, I'll bet you almost all of us have had influenza in our lives, and I don't need to tell you what the symptoms of influenza are, but sore throat, fever, cough, body aches, miserable illness. Parenthetically, when I was an intern in internal medicine at UC Davis in Sacramento, working in the ER, I caught bona fide influenza. Back then, no one wore masks. And I know it was influenza because my attending physician saw me dragging around, said, "You're ill. Let's go draw, it's winter. Let's draw your blood. We'll send it to the lab. Let's get your antibody titer for influenza acutely while you're ill. That antibody titer should be low. And 4 weeks later, I'm going to draw you back here and get another blood specimen," which he did. And it showed a 4-fold rise in antibody titers. So clearly a lab confirmed case of influenza. Back then we didn't have fancy PCRs and so on. It was miserable. I was in bed missing my duties for a week in Sacramento, which meant another intern had to be pulled out of the ICU to cover for me. You feel not only ill, but you feel guilty in my case. So this is a real disease, no doubt about it. Typical duration, 3 to 7 days, longer in older immunocompromised persons, and there is a treatment, Tamiflu or oseltamivir, for both influenza A and B if you're over 2 weeks of age. Okay, let's move on. All right, so if we're to understand bird flu, we've got to speak about the structure of influenza virus. The better we know the structure of influenza virus, the more we can understand about what is happening in the world of influenza viruses, the changes that are taking place. So influenza viruses are RNA. They contain RNA in the center of their core. And they are in segments, in the case of influenza, 8 segments of RNA, each of which codes for 1 or 2 glycoproteins. Those are just proteins, like a stem. You can see that stem sticking out that are topped by a sticky sugar, basically a sugar molecule. So there are 2 main types of glycoproteins with influenza: hemagglutinin, HA or H is called, and neuraminidase, NA or N. The important thing about these 2 glycoproteins is that hemagglutinin allows the influenza virus to attach to a respiratory tract cell, and neuraminidase then allows these newly produced viruses to leave the cell and go infect other people. There are 18 types of hemagglutinin, 11 types of neuraminidase. Okay, and the thing about it, these 2 glycoproteins, hemagglutinin, you can see up at the top left at 11 a.m. and about 6 o'clock neuraminidase, they're changing all the time they're changing, dodging immunity, and that's why we need annual vaccines as humans. Next slide, please. All right, now this slide just summarizes the distribution of the 18 hemagglutinins on the left and the 11 neuraminases on the right. And you can see that depending on the species of animals across the top of each, the distributions in humans is different of the types of influenza viruses than the distribution in poultry, for example, or swine, or bats, cats, dogs. There are different worlds of influenza depending upon the species of animal, which viruses infect whom. Okay, let's go on, let's move on. A couple key terms we also need to discuss and define in the context of influenza and from the standpoint of human infection. So when you hear the term seasonal influenza, that's the annual rise and fall of infections we see every year. This just results from that ongoing process of mutations of hemagglutinin and neuraminidase, those 2 glycoproteins that are on the top of around the outside of the virus. In terms of global deaths per year of humans, they're in the thousands of deaths each year from seasonal influenza. But fortunately, vaccines are made in time to protect us. Let's go over to pandemic influenza. So these are periodic explosions of infections, and they result from major changes in hemagglutinin and neuraminidase. And the number of deaths per year when we have pandemics of influenza is not in the thousands, but in the millions. And vaccines are not always there to protect us because they can be a surprise pandemics. We don't necessarily know. That doesn't apply to this one, H5N1 that we're getting into. We know about it. And that goes back to the first slide, the stockpiles are in place to hopefully deal with it should it occur. Avian influenza is just influenza virus presence in birds, infections. In birds, by the way, in wild birds particularly, influenza hangs out partly in their lungs, but largely in their intestines. The receptors for birds' influenza virus are in the intestines, so they're shedding it in their stool as well as saliva and nasal secretions. In terms of human deaths from avian influenza, very few each year presently. But the key is avian influenza viruses serve as a genetic pool for the exchange of genes with human influenza viruses. And this exchange of genes is what creates constant change in the influenza viruses that we as humans see as well. Okay,
Well,
next slide. Yeah, question.
how do the scientists prospectively predict what the mutations are going to be in order to make a vaccine in time for the next season?
Good, excellent point. So constant, under the coordination of WHO, World Health Organization, constant surveillance by hemisphere of which viruses are circulating when. And so when we see through this surveillance system, new predominance of hemagglutinin changes or neuraminidase changes, that surveillance data can be used toward vaccine production. And vaccine production, three different major ways of vaccine production. Chicken eggs are used to incubate these viruses called candidate vaccines for the next vaccine. or there's recombinant methods, or there's cells. You can actually grow these viruses in cells. But it's through surveillance, to answer your question, that we can predict what is likely to come next.
But, and I hate to interrupt,
No,
but
no, not at all.
the meeting, the annual meeting was called off.
Yes,
And I don't know if it's been reestablished at all.
I don't know either.
So the vaccines for next year are not being considered.
Won't we rely on WHO in that case, World Health Organization, if they release their information?
This is my hope. Despite what is happening in the United States, I'm hoping the brother and sisterhood of science globally will make up, Carol, for hopefully these kinds of, I'm gonna say short-sightedness on our part. So yeah, let's hope World Health Organization is robust and funded by other nations.
And we accept their work.
Yeah, that's a good point. We really need the brotherhood and sisterhood of other nations as we move
Who
forward on this.
produces the vaccine, though? Is it they're privately produced?
They're privately produced. That's exactly right. And so there's still the profit motive, thank goodness, to producing vaccines.
Well,
They're not all publicly.
Right,
funded.
exactly. But the public coordination, we may be a little anemic as we move forward in terms of our public leaders. But again, WHO and our universities, there are good minds out there and in the academic centers. And hopefully the power of truth and science will prevail when it comes to infectious disease control. That's my hope. It could be naive, but that's the way I'm gonna continue to think. All right, so we talked about seasonal, one slide on seasonal, one slide on pandemic, and then we'll really bore into H5N1. Here's data from Centers for Disease Control on the seasonality of influenza, where we see the seasons across the lower axis and the percent of tests that are taken from folks with respiratory diseases that happen to be positive for influenza. What it's telling us is that what we know, influenza rises in late fall, peaks in winter, and then drops off again. Now, there's some interesting finding here from the last 6 years of data for this graph. And it's this line look at that blue line along the baseline. That was when the year COVID broke out. Influenza took a body blow from another respiratory infections in terms of incidence. Almost flat there. In fact, for that year, CDC said the number of hospitalizations for influenza was too low to even estimate. Whereas normal seasons, it's recovered. Influenza is back again. Anywhere from, in terms of hospitalizations, 100,000 to 600,000 a year, and deaths 5,000 to 60,000 a year. Influenza has returned. Next slide, please.
Why would that be?
I beg your pardon?
Why would that be?
Largely because we remember how we segregated ourselves, we all cloistered, and you know,
Mixing, yeah.
we
Yeah.
were.
Here's the stuff,
Okay.
but like I said, there's no paper at all in there. So you'll have to go back to your order.
Okay,
Okay.
one slide on pandemics now. So listed here are 3 pandemics from the last century: Spanish flu on the far left, Asian flu, and Hong Kong flu. And where they originated is unclear for Spanish flu, but southern China for the other 2 pandemics. For each of these pandemic situations, a new virus, novel virus, emerged. H1N1 in the case of Spanish flu, and it displaced the circulating seasonal virus that was circulating around the world, and it took predominance, it dominated. Because its hemagglutinin and its neuraminidase were unfamiliar to our immune systems, we had huge number of cases, we had pandemics. Let's talk about the reproductive number. That's defined as the number of cases that any given infected individual would be expected to transmit to others. How many people would become sick from this person having become ill? For influenza, it's about 1.5. By comparison, measles, much more transmissible, about 12 times as transmissible. And we're hearing a bit more disconcertingly about measles. We shouldn't have this occurring. We should not be having this occurring. Vaccines work. We all know that in this room. But policies are getting tested right now. We'll see where that one goes because of its reproductive number, measles. Getting back to influenza though, estimated case fatality, percent of people who die, high for Spanish flu, lower for the other two pandemics last century, but 0.2% can become a powerful number when you're talking about global populations, billions of people. 0.2% adds up to millions of excess deaths that can occur with influenza pandemics. Curiously, why would some pandemics cause young adults to be mostly affected, whereas another influenza pandemic would be children, and yet another all age groups? One possible answer could be that cumulatively past exposure in life could result in immunity to a new pandemic virus. And that's believed to be the case with H5N1, should we have a pandemic. Those of us, speaking of myself, that are getting on in age, may have been exposed to a number of influenza viruses over our lifetimes, and that residual immunity could be productive. So that's a good thing. And economies take a hit, of course, from pandemics. All right, next slide, please. So I said that, we all know that influenza viruses change all the time, and I like to think of engines, two engines of RNA change. They're called antigenic drift and antigenic shift. Real briefly, as influenza viruses replicate, they're not perfect in replicating. They make mistakes. Those little bases in the RNA, oh, I put the wrong one over here, or substituted the wrong place there. These small changes are called antigenic drift, and they're responsible for seasonal influenza each year. And that's why we need annual vaccines. The other type of engine is antigenic shift. That's an abrupt, major change in hemagglutinin and or neuraminidase. And these shifts can happen, for example, when an influenza virus from an animal population gains the ability to infect humans, or vice versa. A human influenza virus gains major new genes from an animal influenza virus. Either way, you've got pandemic viruses potential there because these new hemoglobins and neuraminases aren't recognized by our immune system, and bang, they can't deal with them, and we got many, many people ill from this new or novel virus. Okay, next slide, please. All right, I want to get now, leading into the second half of the talk, get into the concept of worlds of influenza virus. I know that's not a very scientific term, but it helps me kind of understand. I hope it'll help you understand the big picture view here of influenza. So let's talk about two, what I call traditional worlds. On the one hand, we've got human adapted influenza viruses that are doing their thing back and forth between people. Another world is the wild bird adapted influenza viruses. And they're the source of all influenza viruses found in mammal and avian species. Ultimately, this is the source, is wild birds, the prime original source, if you will. And there are over 100 species of wild birds that carry avian influenza viruses. And they're constantly migrating, right? All these flyaways over each continent, thousands of miles each season, going north, going south, shedding viruses from their GI tract. Those are the receptors for influenza in wild birds, largely through the saliva. Birds produce saliva, and even secretions from the nasal secretions. Birds visit us in marshes, they visit us in shorelines, they visit us as bird feeders in our yard. We do encounter wild birds. We're just maybe not recognizing it all the time. And that's why there's a line between the two worlds of genetic material transmission. One of these wild bird viruses that brings us here today is H5N1. It has earned the designation a highly pathogenic avian influenza virus. Where does this come from, this terminology? Well, it comes from poultry being our standard gauge for the pathogenicity, the virulence, the seriousness of illness that comes along with avian influenza viruses. So if an avian influenza virus in poultry leads to no disease, but infection, or mild disease, it's called low pathogenicity avian influenza virus. If it causes severe disease in poultry, it's highly pathogenic. And that's what we're dealing here with H5N1. Okay, let's move on. This slide just summarizes the number of human infections with H5N1. These are humans now. And the story begins in 1997 in China when this particular virus, H5N1, was documented to cause human infections. And then as time passes, you can see green is Asia, and then it gets into Africa, sort of the magenta color there. And then presently, more new activity, particularly in North America, South America, and Europe. It's moved across the world since 1997 is the bottom line. Next slide. All right, this may be very hard to see, but it's because it's got a lot of data. But I'm going to spend a little bit of time on the slide because it is loaded with lessons for us here about H5N1 interaction with it. So this slide is the history of human, underscore human infections with avian influenza viruses in general. When you see red, that's highly pathogenic virus. Blue, low pathogenic viruses. Most of these infections to humans came from poultry that we're seeing here. And it starts way back in 1959, the data, and it goes up to 2016, the data. So when you see a little stubby arrow, that means that there were human infections only in a short time in time. But if you see the bars, like the red and the blue bars, that means it's continued over the years. That particular virus continued to cause human infections over time, as you see these two bars here and some bars down there. Now, another thing that I didn't appreciate until I really studied this data was that if you look at the different number of combinations of hemagglutinin and neuraminidase, there's 12 different types of combinations here, each constituting a separate avian influenza virus that have been documented to cause human disease. Quite a variety, reflecting all the changes in their mutations that are going on. Back in 1996, H5N1 was found in geese in China. 1996, the virus that we're dealing with now, it was found for the first time, detected for the first time in geese. And shortly after that, in 1997, it caused large outbreaks of disease in Hong Kong poultry and began to spill over into human beings back in 1997, H5N1. It began spreading rapidly through these migratory wild birds, as we talked about, these 1,000-mile journeys by season, spreading across the globe, all the while exchanging genetic material with other birds, the influenza viruses of other birds as it was flying over worlds. And some variants emerged, and I only bring this into discussion. It's probably unnecessary terminology, but variants emerged in genetically distinct groups called clades. You'll see that word clade, just think of group. And specifically within the clades, a further subtyping called 2.3.4.4b. That's H5N1 unless proven otherwise. Two more things about the slide. How many human cases of H5N1 have there been over this period reported? Over 900 according to World Health Organization, worldwide, 23 nations, 52% fatality rate for H5N1. That's a pupil dilator. That's, whoa, 52%. To be honest, these are the most severe infections that come to our attention. Had surveillance been complete globally, we would know about a bunch of milder infections. So the true case fatality rate is probably far less than 52%, but this is the surveillance data presented here. All right, one other thing I want to bring out of the slide and we'll move on. And to me, this may be the most concerning. If we look at 2013, you will see blue, low pathogenicity virus in China detected H7N9. I'd never heard of that one. But then it switches from blue. That's H7N9 in 2016. It's now highly pathogenic. It acquired the RNA changes to go from low pathogenicity to high pathogenicity in a matter of 7 years. Or no, 3 years, sorry, 3 years. Last week, I got chills when I read that earlier this month in Missouri, a chicken flock was experiencing deaths of its chickens, not from H5N1, from H7N9. These creatures, these viruses are circulating. Comes back to that migratory bird mixture of viruses around the world, which leads to the final concept I really want to underscore is you heard the term One Health. I'm sure some of you have heard the term One Health. It's a system of thought whereby we think of diseases like this, zoonotic diseases from animals to humans, in terms of three things: humans, animals, and ecosystems, or humans, animals, and the environment. Those three variables are all at play into everything we're talking about here, beginning with wild birds flying over great distances with, environmental stressors upon them. Marshes are drying up, forest fires are taking away their environments. There's more cattle production for food. There's more poultry production for food. These kinds of animals get infected. So let's move on to the next slide and really delve into this concept of One Health: people, animals, and the environment. But first, we talk about deaths. And WHO is this 52% case fatality. Let's go to Thailand, 2004, where there was probable chicken to human transmission and then person to person transmission. We don't have efficient person to person transmission currently. It is not there, fortunately. But what happened was in Thailand in 2004 was that a 9-year-old girl lived with her 30-year-old aunt. She fell ill after exposure to dying household chickens, became severely ill, hospitalized, and died. Her mom wasn't living with her at the time. From a distant city, though, she rushed to be with her daughter and was in the hospital and provided that daughter 18 hours of unprotected nursing care in the hospital. Mom had no contact with ill chickens, but she died of pneumonia 10 days later. The aunt, who the girl had been living with, provided also unprotected nursing care before she too developed fever and pneumonia, but fortunately she lived. Now, here's the key. Autopsy tissue from the mother who died and the throat swabs from the aunt who lived both yielded H5N1 virus. There were no additional human cases, no further spread, but genetically, the virus obtained from the aunt and mother was closely similar to H5N1 viruses in chickens circulating in Thailand. Bottom line, you can get it from chickens. We now know that. That's second nature now. But also occasionally a human who gets this illness can transmit it to another human. Rare. This virus has not got the capacity yet to be efficiently transmitted. Next slide. All right, we'll start to close now. This is the last few slides here. And I want to get back to that concept of One Health. People, humans, animals, environment. We talked about the two worlds already of human adapted influenza virus and wild bird adapted viruses. Let's start looking at the arrows now. That's where our focus is. How are these connected, these different worlds? We're seeing vividly how they're connected. So for example, H5N1 in wild birds clearly has gotten to poultry. How does that happen again? Think of these flyovers. And the wild birds coming down to feed. If it's on a farm with poultry, could be exchange of virus there. So from poultry, it goes elsewhere, but let's stop for a second. How badly has poultry been affected worldwide from H5N1? Huge number of deaths. Over 160 million deaths globally of chickens due to infection or the euthanasia of chickens to try to get rid of it from the farm. So lots of deaths in poultry. From poultry, it's very likely that non-human mammals have become infected from chickens that happen to be on the farm, for example, or directly wild birds to non-human mammals, not just cows, dogs, cats, but wider and expanding array of species of wild animals now known to be potentially infected with H5N1, raccoons, mink, bears, seals, porpoises, and a wide array of non-human mammals. Now, here's a curious, let's take a little side route here. Our pets can get H5N1, and you probably saw a press release from FDA recently saying, make sure your pet food was manufactured with heat if it contains products from poultry or cows. And there was like 11% of manufacturers that were not heating. They wanted natural kind of ingredients for their pets. And if poultry or cows have H5N1 in the products being used to make pet food and it's not heated, cats and dogs were getting ill. Another arrow there. All right, finally, let's close the loop. We know back in 2022, first time documented in the US, poultry transmission to humans, poultry workers who are culling the birds, euthanizing them, getting sick in Colorado from H5N1, and cows as well. California, I can't get too deep into my work at FDA right now, but California raw milk production is a hot topic right now. 70% of California's 1,000 cattle herds are infected with H5N1. Not all the cows on all the farms, but 70% of the herds in California, cattle herds, are infected. There are raw milk drinkers in America, and it's been shown that H5N1 virus is present, can be present in raw milk. These are major FDA concerns right now. I can't go into them deeper, but just know that this connection here is pretty darn strong. Okay, let's move on. We got 3 or 4 slides and that'll do it. So this slide just summarizes the confirmed human cases in the United States from H5N1 since 2024. On the left side, we see states. Across the top, the exposure source: dairy, herds, poultry farms, backyard chickens, and unknown. Overall, let's go to the bottom line. 68 cases have been reported of H5N1 in humans in the United States since 2024. 2/3 are from cattle exposure, dairy herds, largely in California. 36 of those cases right there. The remainder are from poultry workers on poultry farms. One loner case here is a gentleman in Louisiana who died of H5N1 pneumonia in 2025. He had underlying medical conditions, acquired it largely from backyard poultry in his yard. So one death. And here you just see clinically conjunctivitis, eye reddening with some bleeding on that side. Inflammatory changes over here are very frequent. Most infections are mild, thank goodness, in humans. And hopefully it'll stay that way. Next slide. All right, let's begin to wrap up here. So what changes are required for H5N1 to become a pandemic virus? If it does so, what changes would it have to go through? Short answer, the virus must acquire the ability to transmit efficiently from person to person through respiratory droplets, just like other pandemics. Same thing, efficient transmission by respiratory droplets. For this to happen, H5N1 hemagglutinin must readily attach to receptors in our human lungs, which are used by human adapted influenza viruses. We'll look at where they are in just a second. H5N1's got to get comfortable with the receptors in our lung, and right now it isn't. It's comfortable with a subset of them, but they're in a very special place, as we'll see. So at least 2 mechanisms could facilitate this ability to transmit through respiratory droplets efficiently. Next slide. All right, one is RNA mutations. We've talked about that. RNA is constantly changing. So mutations in viral RNA, which code for hemagglutinin, could allow H5N1 to attach to this specific receptor called, all right, I'm gonna ask you to get familiar with 2 very scientific names: alpha-2,6 receptors. This just refers to something called sialic acid that lines the airway, a little glycoprotein with a sticky surface out there. In our upper lungs, we have this receptor called alpha-2,6, which is what most human influenza viruses attach to. That's what initiates infections. Presently, H5N1 doesn't like that receptor. It likes another one called alpha-2,3 that's located deeper in the lung. Okay, so these deeper locations, though, are hard to get to by avian influenza viruses, because we've got cilia, little brooms sweeping up the germs that come into our lungs, mucus that plugs them up, and immune cells that kill them. Let's go on to the next slide. The second mechanism by which H5N1 could become pandemic, and this is a biggie. This is sort of the holy grail of pandemic routing, if you will. Reassortment involves not little individual changes in RNA, it's wholesale mixing and matching of genes. And if the right mix match occurs, efficient transmission person to person could readily occur. technically, and I'll show you in the last slide or two and we're done, reassortment involves simultaneous infection of a single host, more than that, a single cell in a host by two different influenza viruses that mix their genes to yield a new virus that could be both highly lethal and efficiently transmissible. Let's look at this one. This is really what I find fascinating. Next slide, please. Really fascinating. So here we have, let's say this is our human cell here, say along the airway, for example. And this is how reassortment could produce a pandemic H5N1. So here's a cell, looks sort of like a paramecium, but that's the cell, and there's the nucleus there in blue. Let's say we have simultaneously two different influenza viruses that have infected this cell. Call the first one L. That happens to be an H5N1 virus, which we've got in America. And it's really virulent, 3 pluses, but not so transmissible, just 1 plus that's sort of hidden there. On the other hand, at the same time, we have a circulating seasonal ho-hum variant or a variety of virus that infects the cell. That's could be an H3N2. That's one of them that's circulating normally at this time of year. Inside the cell, these two simultaneously present influenza viruses start to replicate using the machinery of the cell and their enzymes that they have. And so what we're seeing, you see these little squigglies here. Some are red, some are sort of blue. I would call that blue screen. So these are the two individual viruses doing their thing to reproduce themselves. Out come the new viruses. Many are identical to their parents. So we've got L from L, M from M, but we have this also reassortant virus, which takes 7 genes from one virus, and it's got one segment, I should say, segment of RNA from this virus. It's a mutant or reassortant. Reassortments like this could endow the new influenza virus to have 3 plus virulence and 3 plus transmissibility. This is the strongest argument that faces huge political hurdles to say, reduce the amount of H5N1 in our poultry farms and our cattle herds. Do not give it the opportunity to reassort itself in cow cells or chicken cells to produce a potential monster. That has economic, huge opposing forces. And there is one secretary of an agency that's, I will not mention, very close to my work, who has proposed that just let H5N1 be in chickens. Very dangerous kind of talk, in my opinion. Next slide, please. Final slide, this is it. Let's bring it to an end here. So what are the location of these influenza receptors in the human respiratory tract? Well, it's strategic here. The blue alpha-2, 6 receptors, which human adapted influenza viruses like to attach to, they're easily accessible. They're in the sinuses, throat, trachea, big air tubes, as opposed to the receptors that H5N1 likes, presently prefers. Those are the red ones, way in the distant part, deep of the lung. So, in conclusion to this talk, if avian influenza virus is to become pandemic, H5N1 or any other avian influenza virus out there needs to readily attach to the easily accessible airways, those blue ones. And virus isolates from previous pandemics, 1918, 1957, and 1968, suggest that avian influenza virus did just that. It gained the ability to readily attach to these easily findable receptors. Will that happen? Who knows? I'm going to stop there. That's enough. That's enough science, I think, to digest. And let's go on to a discussion. If there's, you know, maybe, what's on your
If I
mind?
need to go back, I will.
Here in the room or on the phone? Yeah.
Yeah, could you speak to the efforts? I know it was an effort in terms of the research space to develop a universal flu vaccine.
Hmm.
Presumably, that's not targeting the sugars because they're way too variable, but maybe the protein backbone. And is that really going to be only effective to the human
Hmm.
you know, the ones, the virus.
Well, in principle,
Families that circulate amongst.
in principle, if you can get sort of low down on the commonality, some mechanism that would prevent influenza infection by some feature of the virus that is universal to all hosts, that is the ultimate answer. Ongoing research, I think there's still a ways to go on
Yeah.
that one. But it's a great concept, a universal approach to a vaccine for influenza, absolutely.
Right, 'cause then you would avoid all every year, we're gonna have to predict
Yeah,
what's going to be widely
exactly
circulated,
right.
as
Yep.
well as
We have
the.
two questions
Yep.
online.
Good point.
Two questions online. One, what agencies are working together to monitor the incidences of?
Okay, what agencies are working to collaborating to monitor the incidence of influenza, you're saying?
Yeah, yes.
Well, globally, WHO comes back to WHO is clearly the leading international agency in keeping track of influenza. But, you know, internationally, WHO surveillance depends upon the rigor of surveillance in individual nations. And that's a very different level of expertise nation by nation. So obviously, the more wealthy nations are going to have more complete surveillance than the less affluent nations, and that can't be overstated. The data is only as good as the surveillance is in the reporting nations. Here in the United States, I like this question because it raises the super important role of local health departments and state health departments. CDC, bless their heart, is a superb agency. CDC is also under the potential scrutiny and budgetary restrictions and philosophical, I'm going to say, pressures of our government, federal government. We, in my opinion, and I've worked at local health departments, I've worked at state health departments, I've worked at CDC. Local and state health departments, in my opinion, are going to have to become leaders in our country. They're going to be loners, but they're got to be leaders. And I have faith in them. They're under pressure. I mean, a lot of states can't really even fund their local health departments very well. But every local health department has a communicable disease section. Every state health department clearly has an infectious disease section. I ran Florida's for 2 years. These are robust groups that are receiving surveillance data all the time. We're going to have to rely on our local and state health departments. Fortunately, they have newsletters you can subscribe to. That means getting 50 of them if you're really interested. But to answer your question, multiple groups deal with surveillance of influenza.
But how about China? China clearly has been the source of a lot of these viruses, and they don't seem to play in the sandbox well.
Very well said, Cheryl. China surveillance, Cheryl makes the point that China surveillance data on influenza, and for that matter, for infectious diseases, I think as a whole, may not be as transparent as, let's say, Great Britain or, traditionally us. Transparency is critical in surveillance, and that's a really good point. There's a gray box there, and I'm afraid we'll be impeded to some extent by the lack of transparency from China, which, as it turns out, has been, I don't want to say home, and this is not a value judgment, but a lot of migratory influenza bird activity has originated in China.
Brigida has a question.
Yeah, Brigida.
Yeah, so thank you for bringing up, the transparency issue. I was going to bring that up because we may be heading into that, that there's a political bend to what we acknowledge. But I had a different question. You mentioned, influenza is a respiratory transmission, but you mentioned that milk is being scrutinized. I understand that they would want to know but we shouldn't be able to get infected through drinking milk, even if cows were to, even if the virus would jump to cows.
Yeah, a number of good points, Brigida. Yeah, let's start with species by species. It all comes down to the presence of those receptors. What type of receptors are where in the body? Cats, for example, are quite susceptible to infection by drinking raw milk from cows that shed H5N1 in their raw milk because of the nature of receptors that allow the cat to become severely ill and die. There have been a number of cat deaths from H5N1 following raw milk consumption on the very farms where the cows are infected as well. Now, it is absolutely true. There is no data, very, very, very sparse data as to the ability of H5N1 through the human GI tract to initiate infection. That is true. The one exception to this would be a publication from out of Vietnam in which two brothers drank raw duck blood as part of a meal, and that duck blood did have H5N1 in it, and one of the two brothers, they both became sick. One of the two brothers died. That's admittedly very minor amount of evidence there, but here's where we got to pull in the physiology that really underscores the need to not be consuming any food that has H5N1 as human beings. In our human intestines, there's a number of alpha-2/3 receptors. Remember, that's the type of receptors that birds prefer to attach to, that avian influenza prefers to attach to. If we allow our human intestines to become hosts to H5N1 virus through raw milk consumption or any other, raw meat consumption, you're giving the opportunity for cells in the GI tract of the human to host not just H5N1, but also human adapted viruses, which attach to the other receptor, alpha-2/6. Those are reassortment possibilities. We as hosts are saying we're willing to take the risk of allowing this virus to reassort in the GI cells, which have both types of receptors. And moreover, do we really know whether a virus, let's say an H5N1 that attaches to a GI cell in a human intestine, cannot gain entry and through lymphatic and blood channels go to the lung? I can't prove this with articles that document that happen, but as an epidemiologist, I say, don't put ourselves in that circumstance. Stay away from it.
Yeah. So if we drink pasteurized milk, do we think that that process is enough to eradicate those viruses?
Without a doubt. And that is really important point. It really doesn't take a whole lot of heat to kill this virus. Pasteurization, without a doubt, makes milk safe. Absolutely so.
Okay.
Now, here's another thing. What about cheese? What about cheese that's made out of raw milk and aged for 60 days? What happens there? Those studies have just been concluded, and they show that cheddar cheese, at least specifically cheddar cheese, that has H5N1 in the raw milk that was used through a 60-day aging process to become cured cheese, this virus has no problem surviving 60 days of aging, no problem whatsoever.
Really?
That data will be published shortly.
Okay, thank you so much.
Wow, I have a question. I live back where geese and ducks walk through my yard. I have a bird feeder that's away from my house. I also back up to water where there are all kinds of ducks and geese there. And there's a path around it that people walk, and clearly the ducks and geese are on that. What is the chance with their defecating in the yard and the ground that people walk on, and then getting into our lungs? I mean, I remember hearing a few years ago, take your bird feeders down, and because you don't want to be, I guess, exposed to what could be around them, or maybe it was just bringing them in. I mean, do you have more information on that?
Well, I mean, the case in Louisiana, I think the bottom line, very, very small risk. If you're asking me for my opinion, very, very small risk of acquiring an avian influenza virus by walking through nature and happening to pick up a virus and make you sick. I think that's exceptionally small. The case in Louisiana, the gentleman who went to his backyard flock and got likely infected there and died, suggests it can happen, but you know, the greater degree of immunocompromised status you have, it increases the chances, but overall, I would stick to don't change your habits of walking through nature and on that account at all. That'd be my take and my lifestyle as well. I wouldn't change it at all.
So, Gloria Andrada, who is a proud California Aggie, she knew that, is, ask us what role can individuals and communities play in preventing and responding to outbreaks of aviation flu.
At this point, I would say keep up with the literature, keep up with your knowledge of biology so that you can make decisions based on science. That's numero uno. Number two is, you know, the obvious, stay away from raw milk consumption. I'm not a believer in it. I think it's a risk. I think it's a risk at this point. And those, I think that, you know, make sure your pet food is properly, you know, produced. And just in general, stay healthy, keep your, you know, your body fit, your mind sharp, and live a healthy life. I know it's very generic information, but that's what I try to do. And that'll take care of not just influenza, but others.
Scares me to death, though.
Yeah, well, that's H cheese, and most producers are not making it out of
Well,
raw
I
milk.
eat cheese because I'm lactose intolerant and I can tolerate if it's been 18 months or more, it's supposed to be safe, so
If it's pasteurized, it's safe. If it's pasteurized, starts with pasteurized, no, not at all.
I'm in France, it's not always pasteurized.
Back to
Yeah.
the question, do you have anything to say about raw oysters?
Oh, raw oysters, you know, that's now in the news because these aquatic avian birds, right, are shedding stool into, but I'm not familiar. That's an interesting question. I am not familiar with any documented transmission of H5N1 through raw molluscan shellfish.
They're like a filter eater, right?
Filter
Yeah.
feeders,
Yeah.
exactly. They can concentrate these organisms. Yes,
Wow.
yes, that's true. But I've not heard of any epidemiology that
One
suggests
more
that.
question, I think this will have to be the
I
last.
heard recently that they had a documented case of airborne transmitted, not fecal matter transmitted, between two chicken farms in Pennsylvania, 10 miles apart. And that they believe, it's a hypothesis, they haven't proven it, but they believe that it's likely that it was airborne.
From farm
From
to
the
farm,
land.
from farm to farm.
From farm to farm.
And they ruled out common feeds
They
or
did.
workers going from one to the other. That'd
Yeah.
be pretty, that's very tough. The diffusion factor would be 10 miles of air.
I don't know.
I can't
It was
refute
a
it.
hypothesis.
Okay, I'll give you that. It's a hypothesis that's worth a thought, but really scientifically rigorous, maybe some more rigor needed in proving that one.
Yeah.
Well, Carl, we can't thank you enough. Please stick with us in our church community because you got us through the pandemic. Now, we're hoping that you'll fend off this thing. And we also really enjoy hearing about your endeavors. Carl is a widely published author and we recommend his books. They're novels, but about all types of social of justice. And we're just so fortunate to have you with us. Before we leave, though, we have Jean Dudek with us right now. If you could just give us a little preview of next week, it's going to be exciting.
And it's going to be very different. from today. The Bible, yes, I'll be talking about two biblical books, the book of Esther, and the book of Judith, which may or may not be in your Bible, depending on whether it has the apocryphal or deuterocanonical books in it. These are two books named after women, but what they have to say to us does not apply solely to women. So I hope you'll come.
Yeah, it'll be exciting. So, you know, check in with us. We're looking forward to it. And thank you all for continuing.
Thank you for helping me. I really appreciate your.
I went through this one dying.
Your,
Yeah. No, really.